Plants are made up of cells. Cells together form tissue, which joins to form an organ. The organs of plants include vegetative parts such as roots, stems and leaves, and reproductive parts such as flowers, fruits and seeds. The growth of plants takes place in two stages, namely vegetative and reproductive. The vegetative stage involves the growth of the plant from a seed to the flowering stage, whereas the reproductive stage is the period from flowering till they produce offspring. During the vegetative stage, the organs of the plant grow in size and number and result in a mature plant with complex body organization. This development of a mature plant involves two processes, growth and differentiation. Now, let's define growth. Growth is an important characteristic that is easily observed in all living organisms. It's a permanent increase in the size of an individual cell in a plant. It requires energy and hence accompanies anabolism and catabolism. In plants, growth is unlimited and hence they retain the capacity to grow throughout their lifespan. This is attributed to the presence of meristems at specific locations in them. Meristems are cells that have the capacity to divide, which add new cells to the plant body. This addition of new cells by meristems is called open form of growth. Meristems are of two types, namely apical and lateral meristem. Apical meristems are located at the root tips and shoot tips of all plants. They are responsible for the primary growth of the plant, that is, elongation of the plant along its axis. Lateral meristems such as vascular cambium and cork cambium are present in woody plants. They are responsible for the secondary growth of the plant, which causes an increase in the girth of plant parts such as the stem and branches. Now, let's see if we can measure growth. At the cellular level, growth refers to an increase in the amount of protoplasm, which is difficult to measure. Hence, growth is measured by different parameters such as size, area, length, and number. For example, watermelon cells increase up to 350,000 times, and hence the growth here is measured in size. In the case of Vittoria regia, a leaf, growth is measured as an increase in surface area, whereas the growth of the pollen tube is measured in length. Similarly, the root apical meristem of maize results in 17,500 new cells per hour, and hence growth here is measured in number. Now, let's look at the different phases of growth. The different phases of growth are zone of division, elongation, and maturation. The zone of division contains the rapidly dividing cells of meristems. These cells are characterized by rich protoplasm, large nuclei, and a thin primary cell wall with more plasmodesmatal openings. The zone of elongation lies next to meristematic tissues. These cells are characterized by enlarged cells, deposition of a new cell wall, a thin cytoplasmic layer, a large vacuole, and a nucleus. Maturation growth is seen on the cells next to elongation tissues. These cells are characterized by mature cells that have reached maximum size in terms of cell wall thickening and protoplasm.